Welcome to the second week of this course. In this week I will explain the properties and capabilities of microorganisms in nature and in industrial reactors. I explain how the basic performance can be quantified, highlight what is needed for scaling up, the use of microbes from lab to commercial scale, and illustrate this with the example of producing the biochemical 1,3-propanediol. Microorganisms are everywhere. In nature, their main goal is to create global element cycles. For example, for carbon, sulfur, nitrogen, and iron. Without full closed-loop cycles resulting in nutrient concentrations and levels that are both life-supporting and promoting, life would not be possible, because key elements would be either unavailable or in toxic excess. Microorganisms, thus, are the masters of sustainable production, and with this I mean the conversion of renewable feedstocks into relevant bioproducts. Humans have domesticated microorganisms for thousands of years to provide healthy, conservable, nutritious and tasty food and beverages, clean water and soil, medicine, fuel, and building blocks for materials. Microbial cells multiply by division and evolve by mutation and competition or selection. The driver of natural evolution is the survival of the fittest in a continuously changing environment. Organisms in an equilibrium state without change will not survive in the long run. This principle can be used to the benefit of mankind to create microorganisms with unique properties, suitable to achieve a certain goal. For example, consortia of different microbes can form biofilms that attach to surfaces like sand particles and thus avoid washout with the passing streams of water. Biofilm technology has been developed in the research groups of Professors Sef Heijnen and Mark van Loosdrecht in Delft, the Netherlands. We can also apply genetic modification and lab evolution under controlled conditions to make cells with specific traits needed for use in industrial processes. And examples are tolerance to high concentrations of toxic products and CO2, and the ability to utilize certain feedstocks at a high rate. Well known is the work done at the Technical University in Delft by Professor Jack Pronk and co-workers to convert the yeast Saccharomyces cerevisiae into a consumer of C5 sugars, impossible for native yeast, while highly valuable in the robust industrial production of ethanol from lignocellulosic material. Cells need a suitable environment to do their work. There should be water, a source of carbon and energy such as sugar, methane or an organic acid, a source of nitrogen such as ammonia, nitrate or amino acids, and further sulfate, phosphate, ions of minerals and metals. Many cells, but not all, also require oxygen. But how much of these compounds is needed? We need quantification. As a start, we could first look at the composition of an average cell. Next to water, which makes up about 75% of the cell, we can distinguish organic macro components such as lipids, protein, RNA, DNA, carbohydrates and fibers, and microcomponents which are metabolites and dissolved in inorganic salt ions. The basis of these compounds are chemical elements, and a typical element composition of biomass, normalized for one atom of carbon, is C1, H1.8, O.5, N.2, and then phosphorus 0.03, sulfur 0.02, and tiny bits of potassium, magnesium, calcium and sodium to neutralize the phosphate and carboxyl groups at the macromolecules, and anions dissolved in the cell. In practice, we usually limit the composition to the elements carbon, hydrogen, oxygen and nitrogen, which cover more than 90% of the dry cell weight. In order to make cellular material, the cell uses anabolic reactions, In these reactions there are always carbon and nitrogen sources, for example glucose or methane, and ammonia and further water, CO2 and protons. Accumulated reactions to produce 1 carbon mole of cells read, for example, 0.175 glucose plus 0.2 ammonia goes to 1 biomass, 0.05 CO2, 0.45 water and 0.2 protons. Or when you go to methane as feedstock, 0.525 methane plus 0.475 CO2, 0.2 ammonia, and that goes to 1 biomass, 0.45 water and 0.2 protons. These reactions require input of energy in the form of ATP. Energy is liberated from catabolic reactions. For example, glucose is combusted with 6 oxygen to 6 CO2 and 6 water. Or 1 molecule of methane is combusted with 2 oxygen to 1 CO2 and 2 water. Electrons are transferred from a donor molecule, here glucose or methane, to an acceptor molecule, here oxygen. This results in a reorganization of the elements from glucose or methane and oxygen to CO2 and water. Other electron acceptors relevant for growth in the absence of oxygen are, for example, nitrate, sulfate, iron 3 and CO2. Further important is to maintain the temperature, pH and osmolarity within certain physiological boundaries, which can vary much among different species who have found and evolved in a niche in nature and became specialists for the local environment. There are extremophilic organisms that grow at hot thermal vents at the bottom of the ocean, in deep mines saltier than the oceans, at 0 degrees centigrade in ice water, or at pH 1 and 13 in acid or soda lakes. However, mainstream would be pH between 5 and 7 and temperatures around 20 to 40 degrees centigrade at moderate osmolarity. This is the basis of understanding and fruitful use of the biochemistry of microbial growth and quantification of the required nutrients and energy sources as well as products. In the next unit we will extend this to the rate of cell growth and the reaction rates of other compounds in a process.